Greetings. This is going to be a quick run through on the answer key to the DNA gizmo. So if you're watching this video, that means you have seen me and we know that you have a completed one. So hopefully you can remember your answers, and this will give you an idea of how and why we have a mark breakdown. So when we look at the first page, the big thing that I needed you to understand was basically the setup and the structure of DNA. So there are a few repetitive questions because they're really important. So obviously when we look at the molecule of DNA, we kind of consider it a twisted ladder, and we call that a double helix. In order for us to replicate it and to copy itself, if you kind of look right here, this is a big giveaway, we have to unzip it. We have to disconnect the two sides. Because remember, this is essentially like a ladder that's joined in the middle. There's a backbone and rungs that meet in the middle. So when you first start doing the gizmo, okay, the two components that are shown within the gizmo itself. Now on this activity, it's called a nucleoside, as well as, a nucleoside, as well as a phosphate. We know that for our intents purposes, we are going to open up that nucleoside, and we're going to understand that that's both the sugar and the phosphate together. Okay, so when we start looking at all of the components that we're looking at, it's going to be your phosphate, your sugar, and your nitrogen base. And hopefully you can actually see those right here in the diagram. The nitrogen bases, again, those are your rungs. Okay, there are 4 of them. There's A, T, C, and G. So this is a really important concept. Make sure you know this. And this is in your data booklet. You don't ever have to memorize it. You just need to know where to find them. So your first practice is going to actually construct a DNA. A lot of you missed this section. This is in fact a please do. So here's what we're looking for. When you actually are given the original strand, so that's the one on the left here, I'm wanting you to make what's called the complementary. So that you are going to be making me the complementary strand. So if there's a T, you're going to remember an A. If there's a C, you're going to put a G. Easy way to remember this is think of apple tree and car garage. Okay, and if you remember that, you're never going to forget what base pairs match up. So A to T, C to G. So you have to put in the right base pairs, but then you also need to put in a backbone. So many of you forgot to do this. Remember, these bases actually need to be stuck to something. So wherever there is a nitrogen base, I need to have my sugar and then my phosphate attaching it. So that's another mark right there. Now that we're on to the next page, you had to make your own. So this is going to be different. I just randomly put some letters together. So the big thing I want you to make sure you did is that you're going to make the original left side, and then you're going to partner it with the complementary. This has to continue with you until the rest of the lab, and I'm going to show you that in just a few minutes. So you're going to see this come back, but again, do you remember A, T, C, and G? And then do you actually know how you're putting this together? So please make sure you understand the sides are your phosphate and sugar. We would consider this the backbone of the DNA. That's another word we're going to hear. And then the rungs, this is what actually goes across the middle. And those are our nitrogen base, or what you would consider a nitrogenous base. That's another way of saying it. So when you actually now start making your replicated DNA from your original, you need to unzip. So the enzyme that unzips it for us is something called DNA helicase. And then, of course, I have my nucleotides. So your nucleotide is the functional unit of your DNA. That is going to be a phosphate attached to a sugar attached to a single nitrogen base. This is considered a nucleotide. So it's super important you understand that. And once I make one nucleotide on the original strand, I need to glue it to my duplicated on my complementary strand. And to do that, we're going to use something called DNA polymerase. You don't need to know the name of that enzyme. Just know that we need to make a glue right here between the two nitrogen bases. And we're actually gluing it using hydrogen bonds. So it's kind of a cool piece of chemistry. And then finally, the last page, or second last page. This is a major error that a lot of you did, so I want to spend some time making sure you understand this. So if I went back to the last page and I had my original, this was the same sequence of letters. Please make sure that when you're copying it, that original is going to create the complementary on the original strand. You should be looking at mirror images. A lot of you simply just coded left to right, and then you just repeated it on the side again. That's not what we're looking for. Is that I tried to do it in color code. So my original was in red, so that was my TACGGCAT. If I were to code its complementary, that's right here. That's the ATGC, etc. When you do that, you have the same sequence, but they're now flipped left to right. So a lot of you miss that. So that's an important distinguishment. So depending on how you did there, you're going to earn a mark out of your perform and record, and that's your skills. Now we start unpacking your knowledge. So can you understand basically the next 4 questions? So these are your multiple choice numeric response, and they're going to show up again on a formal test, but slightly different. So the first question here, and I realize it's small, but I think most of you will be able to kind of see the detail, is that when I take a look at this numeric response, I need to be able to match them. So I need to be able to have what are the terms associated with the proteins. So the sequence should be 2, 4, 1, 3, but then you're going to earn a point or 2 points in the explanation. So don't just rewrite it. Here's what we're looking for. Okay, so if I took number 1, which is an enzyme, this is again something that's going to help a reaction. And we have talked about lots of different enzymes in our body. So this is a special chemical that makes reactions happen faster and better. When we take a look at number 2, this is a protein that helps red blood cells transport oxygen. That's your hemoglobin. So again, just reword this in your own words on what hemoglobin does. Again, another protein. And a section of the DNA that codes for a specific trait or a protein, that's your gene. So again, some of you even drew me the fact that this is what a gene looks like. So I can actually have a section of here's my chromosome and this is a gene. That's a great explanation as well. So you just need to make sure you can explain what a gene is. And then the last one is an amino acid, number 4. There are basically 23 of them. And you actually only make about 17, which means we have to be able to eat the rest of the amino acids through our diet. Okay, so again, this is in our data booklet on page 13. So your sequence should be 2, 4, 1, 3, but again, you're going to earn 2 points for your explanation. For number 2 here, we should be looking at C. So this is again one of these 2 rows. We're in blank number 1. This is the rails. So this is you being able to understand that the rails are the backbone to this DNA. And again, this has been a repetitive concept. And then the rungs, which is number 2, that's going to be the ones that go across the middle. Okay, and even the diagram gives that to you. So you need to know that your backbone is your sugars and your phosphate, and your rungs are going to be your nitrogen bases of, again, A, T, C, and G. So again, your explanation, ideally, I'm looking for, can you reword it instead of just giving me what the question was? And then we look at number 3. This is a good one. This is one of my favorite questions, hint, hint, because you have to kind of tell me what things are not. So the correct sequence is 1, 2, 5. And again, I can put it in any order as long as I have 1, 2, 5. And then you have to take apart what these are. So this is, again, the 3 components of a DNA molecule. So if you were to take it apart, your cysteine, number 1, this is your C, and it's always going to bind to your G, so your guanine. Your 2 is your phosphate. Well, obviously, that's the backbone of your DNA. So we've been drawing those in circles the entire time. And number 5, well, those are my sugars, and we've been drawing those in sort of that pentagon. So this is basically a nucleotide. I know it can't be numbers 3 and 4 because 3 is tryptophan. That is actually an amino acid on your table, so not part of DNA. And a chromosome, number 4, well, that's a package of DNA, so not part of a nucleotide. And last but not least, this is your mutation section. So I'm not going to take you through this too much word for word, because I think the answer key does speak for itself, but I am going to highlight a few things. Number 1, here's your original strand that we've given you in bold. And so your complementary, this is you coding A for T and T for A, etc. And again, I've kind of divided them in 3s to make it easier. Once I have my complementary, that complementary is now going to code for specific amino acids. So if I look at TAC or TAC, that's my triazine. If I look for CCA, that's going to be my proline. And this is just using, again, page 13 in your data booklet if you needed that. Okay. The big trick here is that the 3rd codon, or the 3rd sort of set of letters, is TAA. That is a stop code. You would stop. A lot of you kept going. You actually told me what these other 2 amino acids is. If you did, you actually lost a mark, because the stop code means we stop coding. So if you actually had it done correctly, you would only have written the 3, because after a stop, I stopped coding. And then we had a mutation. We had an insertion, which we added an A basically after our third G there, and we highlighted that. So by doing that in red here, this was basically what the new or the mutated one would look like. That's the original strand. And then I recoded for the complementary in blue, just to kind of help you. Now, by doing this, I still had TAC, which was still triosine. I still had CCT, which is still proline, which is great, even though it was a slight difference. That's, again, that redundancy. But then when we got to our third codon, it should have been a stop code. But it wasn't. It should have been TAA, but it's now CCT. So I now have isosceline. So this is now the wrong amino acid. So because it's the wrong amino acid, the type of mutation that is this is a missense. That's the type. And it's caused by an insertion or a frame shift. So remember, your only three types of mutations. There is a silent, which means I have the wrong coding, but I have the same amino acid. The one that we've just created here, this is a missense, where I have the wrong amino acid in place. And then there's something called a nonsense, where I now have inserted a stop codon in the middle. And these ones are probably the most dangerous. So some of you might need to review this. So I hope this helps. And if you have any questions, see me at Focus.